Hi, everyone. I'm James Bibb from the University of Alabama at Birmingham, and I'd like to welcome you all back to this afternoon's tumor biology session. Tumor biology and physiology is a complex interplay involving cellular heterogeneity and crosstalk that ultimately determines prognosis and treatment efficacy. No two tumors are alike, underlying the need to better understand tumor biology in order to achieve effective patient-specific treatments. In this session, we will explore the latest advances in neuroendocrine tumor biology. This session will also highlight important new cellular mechanisms affecting epigenetic and metabolomic regulation of tumorigenesis in neuroendocrine tumors. As our first speaker, Laura Banaszynski from UT Southwestern Medical Center will present her findings on the role of G-quadruplex or G4 DNA structures and their relationship with ATRX and DAX complexes in exerting mutational burden in NETs. Then, Pawel Mazur from MD Anderson Cancer Center will report on the development of a new clinically accurate PNET model mouse and his studies of the lysine methyltransferase NSD3 as a potential critical mediator of tumorigenesis. In addition to hosting, I'll also serve as the third speaker and will tell you about a novel tumorigenic signaling cascade that is invoked by inborn errors of metabolism that gives rise to pheochromocytoma. Finally, Beatriz Soldevilla Navarro from Research Institute Hospital 12 de Octubre in Madrid will tell us about an exciting metabolomic study she has been conducting in patients to better understand the disease state of neuroendocrine tumors and possible new diagnostic strategies and therapeutic targets for these patients. Thus, this diverse panel will provide exciting new information about the latest advances in NET tumor biology. I'm sure you'll enjoy this session.
Hi, my name is Laura Banaszynski. I am an assistant professor at UT Southwestern Medical Center, and I am very happy to be here today to tell you about some of our NETRF-funded work. So my lab is focused on the histone variant biology. We try to understand the role of the histone variant H2.3 in chromatin dynamics and genome regulation. So we know that this particular histone variant is deposited into chromatin at active regions of the genome. We find it at active regulatory elements such as enhancers. We also find it at promoters and into active gene bodies, and this is all carried out by the chaperone HERE. But over the course of the last 10 years, my lab and others have identified another unique chaperone complex, ATRX and DAX, that's responsible for H2.3 deposition into chromatin. This complex deposits H2.3 at repetitive regions of heterochromatin, for example, telomeres, pericentric regions, and interstitial heterochromatin. This particular complex has become of great interest to the neuroendocrine field because of some NETRF-funded studies that show that ATRX and DAX are commonly mutated in neuroendocrine tumors. Over the course of the last 10 years, the model that's emerged is that the role of ATRX and DAX is to help resolve these G-quadruplex structures at these repetitive regions. G-quadruplex DNA is a non-B form DNA that's thought to be quite stable and is thought to be a barrier for DNA replication. This is evidenced by the fact that cells that lack ATRX show delays in replication, they show replication fork stalling and collapse, and they show sensitivity to compounds that stabilize G-quadruplexes such as PDS. There are a number of helicases that are known to interact with G-quadruplexes in the cells, and ATRX is unique among these helicases in that it partners with DAX to act as a histone chaperone. So the goal of our project was to try to understand the roles of ATRX as a helicase and also its roles as a chromatin remodeling protein, and how that plays a role in replication of these G4 regions. So the first thing that we did was a proteomic study trying to identify ATRX interacting proteins. And what we identified is that ATRX interacts with a number of the MCM helicase complex members. We first used immunoprecipitation followed by Western blotting to validate this result. And what you can see is that from our wild-type lysates, we see interaction with MCM2, MCM4, and MCM6, and we do not see this interaction in our ATRX knockout cells. You can see that this is a substoichiometric interaction, and we think that this observation would be predicted based on the idea that we don't think that ATRX would be involved in all replicating foci within the genome, but perhaps only a small subset of them. In addition to the MCM complex, we also identified a number of G4 interacting proteins, such as DDX10 and SMARCA4, and this again adds credence to the hypothesis that ATRX is important for playing a role in G4 biology. Okay, so the next thing that we tried to do was associate G4 with newly replicating DNA in cells. And here we turn to a PLA assay where we look for G4 interaction with nascently replicated DNA labeled by a short EDU pulse in the cells. And what you can appreciate, I hope, here is that in the absence of ATRX, we see increased G4 EDU foci. And this is more easily visualized in this quantification where we see increased incidence of G4 EDU accumulation in the absence of ATRX. This is phenocopied in the absence of DAX, and this is our first piece of evidence suggesting that the ATRX-DAX complex is very important, not just the helicase activity of ATRX. We next turn to a PDS sensitivity assay. This is a small molecule that's known to stabilize G4 structures in cells. And so the first thing that we did was ask how our ATRX knockout cells respond to PDS treatment using a cell viability assay. And so what you can see here is that in agreement with the literature, that loss of ATRX sensitizes cells to PDS treatment. Further, we phenocopy this result in our DAX knockout cells, again suggesting that the complex of ATRX and DAX is very important. We next turn to the ATRX structure, which is well annotated. First, what we did is we showed that when we add back full-length ATRX to our ATRX knockout cells, we rescue this G4-mediated sensitivity that the cells experience. Next, we asked how particular regions influence this ability to rescue G4-mediated stress. First, what we did is we made two mutants that are known to affect ATRX helicase activity. When we introduce these into cells, what we can see is that these cells remain sensitive to PDS treatment, suggesting that the helicase activity of ATRX is very important. Next, what we did is we asked whether ATRX-DAX interaction was important. And here what we did is we took a mutant of ATRX that is known to disrupt its interaction with DAX. And again, this mutant is unable to rescue sensitivity. And so here what we can conclude is that both the helicase activity and the chaperone activity of ATRX are important for its function at G4 regions. So since our data suggests that the chaperone activity is important, we wanted to take a closer look at the chromatin state. And so here I'm showing you some ChIP-seq browser tracks showing various enrichments at a particular locus in the genome. And what you can see is that this particular locus is predicted to carry a G4 structure. It's bound by ATRX, and it also shows high levels of H3.3 enrichment and high levels of heterochromatin as marked by H3K9 trimethylation. Then the next question that we wanted to ask was how ATRX loss would influence the chromatin state at these regions. And so what we did first was we looked for H3.3 deposition at ATRX-bound G4 regions, and what we see is that in the absence of ATRX, we show reduced H3.3 enrichment at these regions. Likewise, ATRX is required for maintaining high levels of heterochromatin at these regions, as evidenced by reduced levels of H3K9 trimethylation in the absence of ATRX. And so since we saw this reduced level of heterochromatin, what we wanted to ask was whether that was associated with any increased accessibility in these regions. And here we turned to ATAC-seq, and what we found was actually very surprising for us. In the absence of ATRX, these regions actually become quite open. And the reason that this was such a big surprise to us is that traditionally H3.3 is thought to be associated with active chromatin and perhaps promote active chromatin, open chromatin states. But here what we're seeing is that H3.3 deposition is actually required to maintain these regions in a closed heterochromatic state. So because we see that H2.3 deposition keeps these regions closed, we wanted to revisit some of the models of G4 and what G4 does to a cell. So the previous model that I told you about is that G4s actually act as a replication stress, and that certainly does seem to be true. But G4 regions have also been positively associated both with transcription and replication. And in this model that I'm showing you here, supported by several papers, suggesting that these open G-quadruplex regions can actually promote origin activity and increased DNA replication. So we wanted to ask whether or not this is happening in our cells. The first thing that we did to address this was to look at nascent EDU sequencing, where we are looking at levels of EDU incorporation as a proxy for nascent replication. And what you can see is that these ATRX-bound G4 regions show increased levels of replication in the absence of ATRX. Further, we took a published data set looking at origin mapping using a technique called SNS-seq. And here what you can see is that when cells were treated with PDS, that compound that stabilizes G4 structures, that these ATRX-bound G4 regions show increased origin firing. And both of these data suggest that the G4 stabilization actually results in increased levels of replication. So our model so far is that both the helicase and the chaperone activity of ATRX are important for its function at G4 regions. The model would be that in a normal situation, ATRX-mediated H2.3 deposition is supporting nucleosome occupancy and heterochromatin at G4 regions. In the absence of ATRX or DAX, these regions become more open and they are prone to early replication and perhaps subsequent replication stress. So then the question really is, how does that influence the tumor state? And so here, as I mentioned, we know that ATRX and DAX are commonly mutated in a number of human cancers. And this has typically been associated with telomere dysfunction, but what we wondered is whether or not these G4 regions were actually prone to stress in these human patient samples. And so here what we did is we took all ATRX mutated tumors or all DAX mutated tumors in the TCGA cohort, and we asked whether or not regions that are predicted to contain G4 structures contain higher mutation burden. And I think what you can see here is that's true. So in both ATRX mutated samples and DAX mutated samples, G4 regions contain higher mutation burden than scrambled control regions of the genome. Okay, we haven't yet looked specifically at neuroendocrine tumors, but that's certainly the next step here. Just to conclude, I'd like to thank the people who did the work. This work was a NetRF supported project, so thank you very much to NetRF for supporting our work. And this work was carried out by a very talented postdoc in the lab, Yuqing Tang. Aishwarya carried out the computational analysis, and Yuqing was aided in this project by Minhua Li and Ryan O'Hara. So thank you for your attention, and I'll be taking questions at a later time, so thank you.
I think that's a fantastic talk, Laura, and we were all amazed by all this work. I had one question so far that we wanted to know that how much you've learned more, maybe from a functional perspective, about these MCM helicase family members, how they might interact with ATRX DAX, if you think the way they function would be consistent with targeting them with inhibitors to increase chemotherapy efficacy. And that may not work out for what you're thinking, but that came up.
Oh, that's an interesting question. So we don't actually know whether or not there's a direct interaction between ATRX and the MCM complex. We think that this actually does require DAX, because we can see that in our DAX knockout cells that ATRX actually then doesn't interact with MCM. I'm not sure that MCM would be a good target in this case. I'd have to think about that a little bit more. I mean, there are some studies that suggest that these G4 stabilizing compounds are actually good targets, and that they can work with in combination with other chemotherapies. But as far as MCM, I'd have to think about that a little bit more.
Thanks. I have a couple more questions. One was from Yo Vinegre, and he asks, how would you fit ALT initiation in your model? Did you see MRN activation and DSB repair initiation?
Yeah, so we haven't tried to, I don't think we're looking at the best model system to look at ALT. I think that's a great question. I think that there are many studies that suggest that ATRX mutation is leading to telomere dysregulation. But our model, I guess full disclosure here, our model system is mouse embryonic stem cells, which I don't think are the best model for looking at telomeres, and certainly not for looking at alt. Do we see increased levels of damage? Yes, we do. We do see increased damage, and so it's possible that that's playing a role here too.
Don Quayle asked, did you test if dual loss of ATRX and DAX exacerbates the phenotypes of the increased G4 EDU and PDS sensitivity? And she also asked if ATRX loss enhances early replication and replication stress that would mutationally would the mutational burden be high? Would burden be highly susceptible to further mutations along that line? So hopefully some of those things.
Yeah, I'll try to take that quickly. So no, I think that ATRX and DAX are acting in complex here, and we have made the double knockouts, and we don't see any exacerbation of phenotype, and I think that supports that they're acting in complex here. I actually have been considering this myself. So like, could we stress the cells and see higher mutation burden? Like, what if we PDS treated our cells, and then will we see higher mutation burden there? So maybe other environmental stressors would do similar things. Yes, I think that's an interesting idea.
Okay, great. We better be moving over to our next session. Thank you, and please keep your questions up. I'm sure Laura will be happy to engage those soon. Thank you.
Hi, my name is Pavel Mazur. I'm assistant professor at MD Anderson Cancer Center. Today I would like to share with you updates of our research on identification of oncogenic drivers in pancreatic neuroendocrine tumors. Pancreas is the site that is one of the most common for neuroendocrine tumors development. Pancreatic neuroendocrine tumors also represents one of the more malignant tumors. At the early diagnosis, pancreatic tumor has relatively good overall survival. However, this survival dramatically reduces once the tumors start to develop regional, and is particularly reduced when the tumors start to metastasize. That suggests that there is major changes that pancreatic cancer develops over the course of tumorigenesis. And it's not easily explainable by the genetic underpinning of this disease, with three major tumor drivers being identified: MEN1, ATRX, DAX, PA3 kinases, and mTOR signaling being the most common. The complexity of the biology of this tumor, as well as the character of the driver mutations, also limit the availability of druggable targets for development of the therapies. In fact, only one target is currently available for the therapy of neuroendocrine tumors. mTOR inhibitors represent that drug that is FDA approved. My lab is very interested in studying enzymes, in particular kinases and methyltransferases, that are critical for tumorigenesis. With those enzymes being exquisite targets for therapy, studying and understanding their role and function in tumorigenesis is particularly important. What we have analyzed here is the expression of pancreatic neuroendocrine tumor gene expression in the aim to identify potentially overexpressing critical enzymes that drives PANET progression. What we identify is NSD3 methyltransferase. NSD3 is one of the key methyltransferase that methylates lysine K36 on histone H3. As you see here, NSD3 is expressed in PANETs in a very high level, especially comparing to the wild-type beta cells. And this expression is highly elevated over the course of tumorigenesis. When we analyze the wild-type expression in the tumor-adjacent cells and within the tumor with a progressive grade of the tumor, we observe NSD3 to be particularly elevated in PANETs. What we also studied is the levels of NSD3 target, histone H3 dimethylation at lysine K36. This immunoblot analysis here shows this more closely in a more quantitative manner. As you see, normal neuroendocrine beta cells express relatively low levels of NSD3, and methylation at K36 is relatively low. It's increased in PANET tumor biopsies with highly elevated dimethylation of histone H36. What was particularly interesting for us is that we could really observe the aggressive behavior of the tumor. which could be assessed by histological analysis, where you see this highly dysplastic clone emerging from the Panet tumor. As you see, it's associated with the elevated NSD3 expression. All of this suggests that NSD3 could potentially play a very important role in Panet tumorigenesis. We wanted to test that using models that are as close to the human disease as possible. We have generated cancer cell lines from patient-derived xenografts that were passaged in the mice. Those cells were used to generate two populations of cells, control cells and the cells depleted for NSD3. We generated two independent cell lines, and both cell lines express very high levels of NSD3. As you see, we have successfully depleted NSD3 using CRISPR-Cas9 system, and what is important, the depletion of NSD3 also triggers reduced levels of histone H3K36 dimethylation. In this assay, we found that NSD3 depletion triggers very significant changes in the ability of the tumor cells to form colonies, very important ability of the tumor cells. With that, we wanted to test if we could recapitulate that important function of NSD3 using animal model of pancreatic neuroendocrine tumors. As I mentioned, pancreatic neuroendocrine tumors are characterized by the set of three mutations that are most commonly found in patients: MEN1, ATRX, and PNET. We have generated a mouse model that contains all of those three mutations, and additionally, we have generated NSD3 conditional knockout to be bred into this genetic model. We aged those mice and analyzed tumor development. What you can observe here is that the MAP mice develop neuroendocrine tumors and hyperplasia of islets. The green arrows point to the tumors that emerge in the control mice. However, in the NSD3 knockout mice, the tumorigenesis was largely blocked, with a number of lesions and the size of the lesions being very close to what could be observed in normal pancreas. Additionally, proliferation, assessed here by the phosphohistone H3, reveals that NSD3 knockout triggers reduction of overall proliferation in this model. That is very exciting and suggests that NSD3 is a critical element, a critical component of the tumorigenesis in pancreatic neuroendocrine tumors. With that information, we were very intrigued to generate a model in which NSD3 activity would be elevated to recapitulate what is happening in a malignant pancreatic tumors that we know express very high levels of NSD3. We have identified point mutations of NSD3 that have capabilities to be more active than the wild-type form of NSD3, and we use that feature of those point mutations to generate an animal model in which hyperactivation of NSD3 could be assessed. We breed those mice into our genetic model of panels with MEN1, ATRX, and PTEN knockouts, and we age those mice as we did with NSD3 knockout. However, the phenotype couldn't be more different. In fact, NSD3 point mutation triggers dramatic acceleration of tumors, with the multifocal tumors of enlarged size, increased grade, and increased proliferation, as you see on the lower panel with the phosphohistone H3, suggesting also more malignant and more aggressive behavior. And indeed, that is the case. NSD3 point mutant also leads to development of metastases in liver, as shown in the picture on the right side. That all suggests that NSD3 is governing a very important oncogenic program that facilitates this transformation and this progression to a very aggressive tumor. We're eager to investigate that, and we collected tumor biopsies from both control mice and NSD3 hyperactive mutant mice. As you can immediately observe in this immunoblood, that leads to increased levels of histone H3K36 dimethylation, which is a substrate of NSD3, which suggests that potentially we will deal with the massive changes in gene expression. And that is a fact. When we analyze gene expression changes in RNA-seq experiment, we identify hundreds of genes that are misexpressed in NSD3 hyperactive mutant. When we try to understand what those changes could mean in terms of signaling pathways, it became more and more obvious that NSD3 triggers a whole oncogenic program, which includes increased activity and increased expression of MYC targets, mTOR targets, and E2F targets, all of which are very characteristic for very aggressive tumors. To validate that, we also used the cell lines isolated from the mouse models to delete the endogenous NSD3 and reconstitute the enzymatically proficient or enzymatically deficient form of NSD3. What this experiment shows is that NSD3 deletion and reconstitution with the enzymatically deficient form of NSD3 is not able to rescue the expression of the target genes like MYC or PRKAA2, which is AMPK target. Whereas the NSD3 hyperactive mutant is able to restore this high expression of those genes, suggesting that indeed the enzymatic activity of NSD3 is required to drive this oncogenic program. That is particularly important because though the NSD3 inhibitors are not yet being developed, enzymatic activity of enzyme is the critical point for development of inhibitor and a potential therapeutic application of this finding. With that, I would like to summarize that NSD3 represents a potentially important epigenetic driver that is triggered by genetic mutations in neuroendocrine tumors like MEN1, ATRX, and PTEN, but drives very consistent and very specific oncogenic program that includes upregulation of MYC targets, mTOR targets, and proliferation genes that is required for accelerated tumorigenesis with a malignant characteristics and phenotype. With that, I would like to thank my collaborators and everybody working on this project, in particular Dr. Orghazani from Stanford University, my lab, and multiple collaborators from clinic and from basic research that took part in this study. I would like to thank Neuroendocrine Tumor Research Foundation for providing support for this study, and I would like to thank you for your attention.
Hi, Pawel. That was just an outstanding talk. Thank you. We all enjoyed that one. So I had several questions that came in. Daniel Chung says it's absolutely wonderful work, and he was asking about the correlation. I think you showed one between NSD3 expression and the state. Oh, he wants to know if it links to stage of PNETs. And we also, many people are wondering, like. Now, what are the mutations that we see in PNETs that have been identified in NSD3, if any? You're talking about an activating mutation. I don't think it was. We didn't catch whether that was actually a human mutation or not. And also, how's this work? How do you end up upregulating NSD3 in the disease? Several questions.
Yeah, absolutely. No, that's an extremely good question. Questions that we ask ourselves as well. So let me start with the expression. So we observed definitely elevated mRNA levels and protein levels when we do stainings. There is we don't have a very strong in terms of numbers panel of pathology of tissue sections to really provide an answer to in terms of progression. If that correlates, increased expression correlates with the grade of the tumor, it definitely trends this way. The activation and what's the mechanism of the activation, I'm not really able to provide you direct answer. There's several of methyltransferases that methylate H3K36. Including NSD2, NSD3 being one of the more active. Why is it specifically NSD3 in neuroendocrine pancreatic tumors? We don't have answer to this question. If in case in your question about the point mutation, so that's the whole story about the point mutation. It is we didn't find this point mutation specifically in pancreatic neuroendocrine tumors. However, we found multiple recurring mutations in this site and few other residues generally in human tumors. And that's due to relatively low number of cases that a sequence of publicly available sequences is there. There's several residues in NSD3 that lead to hyperactivation, which is whole new finding. It's relatively uncommon, very rare to find a point mutation in metal transferase that would lead to hyperactivation. So that's also a kind of exciting news for us. We're also studying like biophysics of this point mutation. We think that point mutation leads to different activity of the autoregulatory loop of this metal transferase. This metal transferase that provides a very kind of elegant explanation why this leads to hyperactivity of this enzyme. I think I covered that
was wonderful. My message board is lighting up with questions. A lot of enthusiasm about your talk. So I'll pick just a couple more. One more actually. So how do you think ATRX DAX activates NSD3?
So we see that effect of NSD3 upregulation in our model. We see that spontaneously over time we see this NSD3 upregulation in the course of the tumor genesis. Those mice that the MEN1, ATRX, and P10, they usually don't develop really very high metastatics, very high grade lesions. However, in those that were aged to very late stage, we tend to see more NSD3 upregulation. For example, in a model which we know that not fully recapitulate what is happening in a human disease, the RIP-TAG model with the mutation of 53 NRP, we actually don't see that effect. So kind of leads me to speculate that it is more specific to those particular mutations. However, the mechanism or how exactly that would be remains still open.
Thanks, Pawel. You're doing great work. Thanks to everyone for such great questions. Unfortunately, we got to return back to this session, but I'm sure Pawel will be able to go through those and answer those for you. They're great questions as well. Thank you.
Thank you. Thank you so much.
Hi, I'm going to tell you now about some of the research we've been doing on neuroendocrine cancer mechanisms and tell you a little bit about some of the animal models that we've been able to develop. So our work on neuroendocrine cancer started when we sort of serendipitously discovered that the aberrant activity of this protein kinase CDK5 could drive the formation of neuroendocrine cancers of the thyroid, medullary thyroid carcinoma. And we reported this some 7 years ago in Cancer Cell and showed that this is a novel way and a novel pathway by which these types of cancers could be caused, and this opened up a whole new way of trying to get at some of these cancers. So more recently, just in August, we published a paper in PNAS where we showed that we can detect CDK5 as well as its activating cofactor P35 in every single type of neuroendocrine cancer we've ever surveyed, and that all the cells so far that have been derived from neuroendocrine tumors have expression of CDK5 as well as its activating cofactor P35 and P25. P25 is the product of calpain cleavage of P35, and it's what causes CDK5 to have aberrant activity. We also found that all these cells so far that we've looked at, all neuroendocrine cells we've looked at, are vulnerable to inhibition of CDK5 as a mechanism for either stopping their growth or causing the cells to die. And in this most recent paper, we were able to find new ways to deliver a CDK5 inhibitor into tumors using leuko-like particles. We were able to identify biomarkers and make phosphorylation state-specific antibodies to those markers, and then use that to predict which neuroendocrine cancers would respond to CDK5 inhibition and which wouldn't. But the rest of the time today, I want to talk to you about some new research that we haven't published yet that I'm very excited about. And this is mostly mechanism-driven research that is focused on pheochromocytoma. And this is the study that we're really working hard now to finish and publish the work on. Pheochromocytoma and its sister disease, paraganglioma, it's characterized by the oversecretion of a hormone, norepinephrine, and this causes problems with various physiological symptoms, and there really isn't a very good effective treatment for this disease, and it has been understudied and undercharacterized. This disease is often considered to be an inborn error of metabolism. That's not necessarily always the case, but the worst cases of this disease are arising when you get mutations in a group of genes that are involved in regulating metabolism. And the most problematic of those are the series of genes in the succinyl dehydrogenase family, SDH, and the ones that are most problematic within that group are SDHB. If you have mutations in SDHB, you usually, by definition, lose function. This causes an accumulation of succinate and leads to problems with pseudohypoxia. Cells think they need to be dividing when they shouldn't, and this is the classic Warburg effect in motion. So as a starting point, we obtained tumors from a number of our colleagues and evaluated them and found that these tumors, which had SDHB mutations, lacked expression of SDHB, normal expression of CDK5, but very high levels of P25. And so we wondered how these things might be connected. We used a human cell line called HVO1 that was derived by my colleague Hans Gehi. And in those cells, we knocked out SDHB, and we saw an increase in P25. When we look at those cells and compare the wild type to the knockouts, we find they have the classic shift over to dependency on glycolysis and an increase in succinate. Because of these problems, the cells are unable to manage calcium properly. And as a result, they can't respond and recover from an insult to calcium, such as adding the calcium ionophore ionomycin. And this effect is mimicked in those cells, not just by challenging with calcium in the knockouts, but by adding succinate. Succinate tends to cause loss of calcium control. Succinate is driving the activation of calpain, and it's causing the conversion of that CDK5 cofactor P35 to P25. So we can say now that SDHB mutations are causing an increase in succinate, which is causing dysregulation of calcium, which is causing aberrant activation of CDK5. Now, we used those cells that Hans had derived, and we began to look at what those look like in xenograft tumors, and we found that the SDHB knockout cells generated a much higher level of P25, had much more activated calpain than normal cells, so that kind of supports our hypotheses. But it doesn't prove that CDK5 is actually causing these tumors. And to do that, we used this approach where we made an inducible P25 GFP overexpressing mouse, and we used the PMNT promoter to drive the expression of P25 in adrenal medulla chromaffin cells. And very quickly, within 3 weeks, these animals started to show signs of these tumors. And the animals go on to develop these tumors. You can image them with DOTATOT, so it closely mimics the somatostatin receptor expression and detection that can be used in humans. They showed the classic endocrinological signatures of increased norepinephrines and an increase in systolic blood pressure that characterize and are diagnostic for this disease. And so we asked, well, if CDK5 is downstream of SDHB mutations and it can drive the formation of these tumors, what might be some of the mechanisms that are downstream of it? In the study we just published, we conducted a phosphoproteomic screen, and we looked at phosphorylation sites that were likely tracking to CDK5, and we looked at those that went up, and we identified biomarkers that were CDK5 substrates that were likely contributing to tumorigenesis in neuroendocrine cells. However, there was also a large group that went down, and we were interested in the phosphorylation state of these proteins going down because they might denote tumor repressors that are being relieved from their ability to repress tumors. And so we started to look at those first, and we screened through about 35 pathways that were significantly reduced in growing versus arrested cells. Some of those pathways such as this protein AAK1, when we mutated the phosphorylation sites to D or E, it really didn't stop the cells from growing. So that really isn't a tumor repressor pathway in these pheochromocytoma cells. However, one called PRKAG2, it was responsive in that if we mimicked the phosphorylation, the cells would stop growing. So we then went and made phosphorylation state-specific antibodies to the PRKAG2 protein, and we found that it was also reduced in human pheochromocytoma cells. And the thing, what PRKAG2 is actually the gamma regulatory subunit of AMP kinase. AMP kinase is an important metabolic sensor which is activated by the presence of AMP. And if in some way it's not activated by AMP, it can release cells from senescence and cause them to grow. And what we found was when the phosphorylation of this site we identified on this regulatory protein, PRKAG2, was lost, we also saw the loss of the activation state of AMP kinase as detected by this other phosphorylation site, threonine 172 on the catalytic subunit. So we have a phosphorylation site that was going down as we were activating CDK5, and we wondered how that could be. And what we ended up finding is that's because when CDK5 is activated with the formation of P25, it induces the inactivation of another kinase that it associates with, GSK3. GSK3 then can no longer phosphorylate PRKAG2, and as a result, the AMP kinase holoenzyme is inactivated. We wanted to target this, and so to target it, we made we screened and identified a number of CDK5 inhibitors. These are novel compounds, and we found that one of them in particular, called MRT3007, was particularly potent and arrested the cells from growing. It was much more effective at arresting cells if they expressed P25, and it only inhibited CDK5, not CDK1 and 2 inside cells. We also found that we could arrest these cells by activating AMP kinase to overcome the loss of activity that we found in our cells. And so we can use various AMP kinase activators, either alone or together with this novel CDK5 inhibitor, to stop these cells from growing. Well, will this work as a therapy? And to look at this, we did a number of control experiments to show that the pathways were activated in the direction that we predicted they would be. But eventually then we gave MRT3007 first to cells, and we found indeed we saw a loss in the inhibition of GSK3, an activation of PRKAG2, an AMP kinase, and a marker also ACC that marks the AMP kinase activity. So it was working the right way in cells, but we wanted to make sure we didn't kill the mice with the doses we were giving, so we defined its toxicity and chose a dose 10 times below the lethal dose. 5 mg per kg when we treated animals that had these xenograft tumors. Indeed, it stopped those tumors from progressing, while it didn't stop the animals from cause any serious toxicity effects. The animals maintained the same body weight. And when we look at those tumors, it activates the signaling cascade just as we predicted it would. To make sure this was a potential therapeutic approach, we've used another model system that Carol Patrick and his colleagues have developed. This is a mouse model of pheochromocytoma, and in this case, treating the animals with this novel drug, MRT3007, did a great job arresting the tumors and may have also started to regress those tumors. So we think this is an exciting new treatment that we might be able to bring to this disease. So to summarize, we've delineated a cascade by which SDHB mutations drive up succinate, triggering loss of calcium control, activating calpain, converting CDK5 to its aberrant form, which then causes the inactivation of GSK3. As a result, GSK3 will not allow the AMP kinase holocomplex to be maintained in its active state, and we believe this is an important contributing factor to the progression of these PC tumors. That's all I can tell you about, except I wanted to mention, because I know that there's a large interest in this audience in pancreatic and GI neuroendocrine tumors. We also have a novel model of pancreatic neuroendocrine cancer that we've made. We have some novel mechanisms that we think are driving this, and maybe I can talk to you about this other model some other time, and I'd be happy to talk to you about it offline. So other than that, our goals are to model and study nets, to develop new diagnostics and therapeutic approaches, and to characterize the mechanisms and develop potential drug targets that can advance this field. I want to thank the people who have been involved in this research. Priyanka Gupta is the one working on pheochromocytoma. Angela Carter has led in the studies of biomarkers and how to target neuroendocrine cancers using that as a guide. One of my clinical colleagues, Sushant Reddy, has been wonderful at helping us to police up tumors and giving us the key clinical perspective and making sure we color within the lines. Corinne Pozo is a former postdoc who did some of the original work. Hans Gahey and Carol Patchik have been just great collaborators, and I want to thank them all, and I'd like to thank you for your attention.
Thank you very much, James. That was a beautiful talk as always. Very, very insightful understanding of the pathways of what CDK5 is regulating, and now in this new model. So great job. Before I go any further, I see a question from Dan Chung. Great talk. Any thoughts on what upregulates CDK5 in PNETs and GI nets? And does GSK3 also regulate Wnt signaling in this cascade?
GSK3 also regulates. I don't know about GSK3 altering Wnt signaling at all. I'm sorry. As far as what upregulates CDK5, it's a funny thing. You know, it's not actually a change in its expression. It's a change in its specificity that arises as a result of calpain activation and driving CDK5 over to this aberrant form where it becomes more soluble. It may enter the nucleus and it phosphorylates a whole bunch of proteins. And that's not a good thing, obviously. It is some of the proteins it may phosphorylate are like RB protein. It wants to play the role of just another cyclin-dependent kinase. But others seem to be somewhat unique and related to the loss in calcium balance. And neuroendocrine cells they're very calcium functional dependent. They have action potentials. Calcium floods in. It causes this whole exocytotic, endocytotic cycle, and so calcium's everything to them. So it's not surprising they'd be vulnerable to some aberrantly activated by calcium cyclin-dependent kinase. So that's the main thing. In PNETs, what are the mutations? It seems like many mutations can get you in trouble with calcium regulation. And I guess, you know, maybe the genes may be quite specific, but there's many ways of getting to this problem. And once you get to it, you've got a kinase that in neuroendocrine cells is particularly dangerous. So I hope that makes sense.
Yeah. So Emily Bergslund also has a question for you. She says, beautiful talk, James. I'm sorry if I missed this, but is this pathway assumed to be relevant only for SDHB mutations? And if so, yeah, go ahead.
I think this pathway is relevant anywhere where you've got hypoxic conditions that are driving loss in calcium management. I think that's probably many, if not most, neuroendocrine tumors. It's obvious that once we put our cells in a tumor environment where things tend to be more hypoxic and more acidic, that the pathway gets amplified. Right now we're playing with hypoxic conditions, but I think that the aberrant activation of AMP kinase may turn out to be common to many tumors. We haven't blotted enough tumors with that antibody yet to say it's beyond just a few others.
Okay. She was also asking about other SDHX mutations and whether they would be similar vulnerabilities.
There is. They all have more loss of calcium. They're all producing more succinate. The SDHBs in our hands make the most P25, and so that might link to some feature of why they're viewed as more fearful from some perspectives. But I think they're all pretty bad.
Okay. I've got a question for you. We're going to have one more question, and then we'll go to the next talk. But if you were to pair your new drug with another drug, what would you pair it with to get the best tumor regression possible?
Well, right now we're using low levels of CDK5 coupled with what I guess is pretty high levels of AMP kinase activators, and that seems to be working. I've also realized that there's actually a succinate receptor. I can't even remember the name of it. It's GLP. It's in the endoplasmic reticulum, and it's a calcium conductor. So there is one antagonist we've seen, and we're going to buy it and try it. And so the idea that you could hit upstream to stop succinate from messing up calcium, and then hit CDK5, and then go downstream hit AMP kinase is a reasonable thing. We know that there's also aberrant HIF signaling. Art showed that, and we're working with a colleague, Erwin Van Meer, who has an actual HIF-1 inhibitor, HIF-1A inhibitor. And some of you, if you're interested in playing with a HIF-1A inhibitor in your particular model, I can put you in contact with Erwin. He's a good scientist.
Excellent. Thank you so much, James. And I think we are ready for our last presentation.
I'm Beatriz Soldevilla, postdoctoral researcher in the Clinical and Translational Oncology Lab of 12 Octubre Hospital from Madrid. And first of all, I would like to thank to NET Research Foundation for giving me the opportunity to present our preliminary data about metabolomic profile of advanced neuroendocrine cancer patients. Neuroendocrine tumors, as you know, are a heterogeneous family of neoplasm of increasing incidence. In the last decades, there are particular tumors, and one of their singularities are that they are able to secrete hormones to the bloodstream. These tumors are called functioning tumors, and this feature makes them optimal candidates for metabolic studies in non-invasive samples. Metabolism dysregulation is one of the key hallmarks of cancer, one of the most emerging. The oncogenic signaling pathways fuels enhanced cell proliferation through cell metabolism reprogramming, and this metabolic rewiring supports tumor development, progression, and metastasis. In order to identify these metabolic changes, metabolomics is a powerful tool to this fact, and also to discover diagnostic, prognostic, or predictive biomarkers, as well as novel therapeutic targets. This approach has been used in other tumor types, such as breast, colon cancer, but the metabolic profile of neuroendocrine tumors has not been described yet. So our main objective is to characterize the metabolic profile of patients with advanced neuroendocrine tumors in non-invasive biofluids. So specifically, we have used blood from a discovery cohort with 77 NET patients. You can see the main clinical pathological characteristics of these patients. We have also used a control cohort with 68 no-cancer patients, and the metabolites from the plasma of all these patients were extracted by standard protocols and analyzed by chromatographic separation techniques, such as gas or liquid chromatography, and also by no-chromatographic approaches, specifically capillary electrophoresis. These three techniques were coupled to mass spectrometry to try to identify the number of the specific metabolites. So you can see in the plots that we have identified with the three techniques a set of metabolites able to discriminate between NET patients in red from no cancer individuals in blue. Here you can see the number of metabolites detected by each technique. We have detected about 1,000, but only 500 significant difference between the groups. And after that, and using public databases and the biochemical characteristics of the metabolites, we have identified 155 metabolites, 14 of them by several techniques. When we classify these metabolites, taking account their biochemical characteristics, we have found that NET patients are enriched in carbohydrates, fatty acids, glycerophospholipids, steroids, and amino acids. To sum up all the information from the set of metabolites, the identified metabolites, we have found that the NET patient present increase in the peptides, also an increase in oxidized lipids, and oxidized compounds derived from arachidonic acid. Also, we have found decreased levels of carnitine and derivatives, and also we have detected a strong dysregulation of the metabolism of arginine, tryptophan, and glucose. So, to analyze the influence of these metabolites in the regulation of the pathways in the tumor, we have used two analyses. First of all, we use published data from the literature, and also we have analyzed this set of metabolites by the MetaboAnalysis platform, and we have found that NET patients are enriched in classical cancer pathways such as apoptosis and cell cycle. Also, they are enriched in NET signaling, in pathway related with NET signaling, for example, tryptophan metabolism, angiogenesis, or mTOR. And also, we have identified novel enriched metabolic pathways related with TCA cycle and also with the metabolism of arginine pyruvate agglutathione compounds. So the second goal of our work was to try to discover potential biomarkers. For this, we have two analyses. In one hand, we have the supervised model previously built. From this model, we obtained the Bibb score, which explains the influence of each metabolite in the separation of the groups. Using this criteria, we have selected 17 metabolites with a Bibb above 1. And for the other hand, we have performed a ROC analysis where we have calculated the area under the curve value. We have used 0.85 as cutoff of this value, and we have selected 5 metabolites as putative potential biomarkers. Also, we have selected 27 metabolites which met both criteria. So finally, we have 49 metabolites with potential biomarkers, which are very important because they are able to clearly separate the NET patients from the non-cancer individuals in unsupervised clustering. So to conclude, we provide for the first time a comprehensive metabolic profile of advanced neuroendocrine tumor patients. We have identified a reduced set of metabolites of potential diagnostic utility, and also we have revealed new enriched metabolic pathways that may open new avenues for clinical research, including potential novel targets of therapy. Thank you very much for your attention.
All right, I just wanted to mention that unfortunately, with apologies, Beatriz was called away at the last moment, and she's not going to be able to be here to answer any questions. But she promised she would be able to answer the questions if we would post them very soon. So please be sure to ask questions about that talk. It was pretty cool.
Jane, I have a...
Yeah, go ahead, please, while I'm trying to figure out what's the first thing.
Great. No, I thought there were a lot of great talks this session, and I was, particularly interested in the talks related to some of these, epigenetic mechanisms that may underlie these tumor formations. We've struggled a lot trying to understand, why there are so few mutations, traditional mutations, and I think that there's a lot that seems to point to the epigenome. So I had a question for Pawel, actually. Your work about the histone methyltransferases is really fascinating and exciting, and it harkens back to work I think Matt Meyerson developed 15 years ago, really, about the relationship between menin and methyltransferases. And I wonder if you have looked to see, and of course I don't think he identified this particular one. I wonder if you've looked to see whether menin, there's any relationship in terms of any protein-protein interactions between those two proteins and correlation with menin mutations, any N1 mutations in the tumors that you see, the high expression.
Mm-hmm. No, you're absolutely right. There's a very, you almost feel like that there must be this epigenetic underpinning of that, and that's why our search for what it could be, what could be like an epigenetic oncogene, if you will, And NSD3, it's interesting because obviously that opens a lot of regions of chromatin, but it's not necessarily very distinctive. Like it's not discriminating where precisely it will open these areas of chromatin. So it kind of opens it to evolution, kind of to start mutating, start expressing multiple different genes, and that's how we kind of look at it. When you specifically ask about the link between MEN1 and methyltransferases, obviously that's a whole big region of investigation. NSD3, perhaps there's this link in terms of expression of NSD3. Why is it this one? There's at least 4 or 5 different enzymes that could do potentially very similar job. ASH1L, NSD1, 2, 3. There's several enzymes playing the same function. I think that there might be some specific link to MEN1, but right now our only suspect is actually histone H3 3.3 or H3Z. That seems to be seems that there is a certain preference to this histone for NSD3. We still pending investigation, but there's in vitro assays that there's definitely a bias towards it. So maybe that's the link. But that's as far as I can speculate right now.
I'm sorry, are you saying that you have higher activity on H3.3 from NSD3?
Yes.
That's a biochemical assay?
That's in biochemical assay. Yes, in biochemical, yeah. Yes, so we purify specifically the NSD3 protein and make the radioactive in vitro. Mm-hmm.
There's a new structure of SETD2 with an H3.3 tail, so that might be useful for you to look at.
Yeah, exactly, right. There's a very unique, there's a very unique difference between NSD2 and NSD3, also in regards to the preference. Seems like there is a slight. We also made a structure of NSD3. There might be actually a very distinctive difference between how those two, NSD2 and NSD3, behave on H3.3. There is a kind of a small, on the C terminal, a small fragment that seems like that provides a preference to this variant. That's also something on our agenda to study. But that's a very good point. Yeah.
Did I miss this? Do you have the genomics for NSD3 enrichment?
So we've made only a couple of studies so far. We did the ATRX, just a very broad characterization of what's happening. And right now we're doing a cut and run for analysis.
NSD3 itself.
Yeah.
Pawel, you mentioned while we're on it, you mentioned that you didn't think there were any NSD3 inhibitors that would come in handy to exploit your discovery. But I have to say, I came up with one where BIX 01294 inhibits all 3, and then there's one called newly released BI 9321, potent selective for NSD3. So maybe, I mean, those could be fake news, but maybe maybe there is something used. It's so you got to be dying to do it.
Absolutely. So the very specific that you mentioned, it's pretty unique. It's a probe, but it is interacting one of the domains. It does not those inhibitors that you're referring to. They're not inhibiting actual enzymatic activity, but the inhibitors, as far as I know, not published, are coming for sure. So there's definitely a big interest. NSD3 is a highly upregulated methyltransferase in one of the big area is squamous cell lung cancer and breast cancer. And there's a huge interest from to develop inhibitors for NSD2. On the other hand, a lot of big tumor relevance as well.
Well, I've got a question for you, and then hopefully we'll spread it out to James and Laura here. I was curious about the tumor types that you were developing. You said that you, in your MAP mouse, you developed peanuts. Are they insulinomas?
Insulinomas, yes. Yeah.
So they're functional. And then in your mutant animals with the activated NSD3, are those non-functional or are they functional?
Yeah. So they seem at least initially that they all functional. However, over time, I'm not sure anymore. It seems like the NSD3 also introduces a lot of heterogeneity in those tumors. So there are like very, very big tumors within the pancreas. Within pancreas, there are still many small tumors that you would probably say, oh, they look very much like in the control mice. So it seems like this NSD3 plays this epigenetic mutagen function.
Yeah.
It opens a lot of possibilities, and not all tumors take the same option probably. So not all of them are the same in terms of also levels of expression of like insulin or also specific marker for neuroendocrine.
Okay. Thank you very much.
Yeah.
Laura, so your work is beautiful, and we've been struggling to try to understand ATRX and DAX in neuroendocrine tumors, and the biochemistry that you've shown us is really fantastic. And it always comes down to, in the end, so how do we link it to the tumors and the tumor formation? And I wonder, and I know that's obviously a big jump, but I'm curious if you've thought about, like, in terms of the known mutations that have been documented in these tumor type, how you think those types of mutations are affecting, the pathways and functions that you're elucidating.
Mm-hmm. Yeah, I think that's for us the struggle too, is trying to go from the biochemistry to the tumor biology. It's always the struggle. And not having the right model systems, I think, is also problematic, because I think a lot of this is really context driven. And probably will be context driven. Many studies have shown that there's alt in ATRX truncal tumors, and I'm sure that's playing some kind of role here as well. We tried, we when we're looking for, we just thought, okay, like maybe we can look at the mutation burden in the tumors, which is what we decided to do. We actually didn't include the neuroendocrine sample in our analysis, because we just made a cutoff of having 20 samples or more, because we were trying to get statistical relevance. Probably a lot of those DAX mutations are neuroendocrine tumors. I would have to go back and look at whether or not they are. I think, I guess I could ask you if you think that that increase in mutation burden is real. It's definitely statistically relevant, it's statistically significant, but whether or not you think that is functional, in the next step that we were trying to do, we've taken it and we've broken it up into different tumors, and we definitely see that across different tumors this holds, with the exception of actually skin cancer. So we don't see an increased prevalence of mutation burden in G4 regions in skin cancers that carry ATRX mutations, but in all other tumors that we've taken out of the cohort, we do. The next step that we were going to try is looking at different kinds of mutations, not just mutation, but different kinds of mutations, and then trying to link them back to whether or not they're proximal to particular genes. I guess the question is, is this a mutation issue? Is this a transcription issue?
Right.
Yeah. Could I jump in there with a question? It's just the whole process of what you're discussing, leading to replication stress, basically. It just crosses my mind. So many other therapies are trying to build on that in other tumor types, where if you have that replication stress and you've activated checkpoint pathways, check 1, check 2, if you come back with inhibitors of those pathways, like check 1 inhibitors, and you combine that with CDK activating drugs, you can push the cells into mitotic catastrophe, basically, and really wipe them out quite effectively. I work on CDK inhibitors, but there definitely is that context where you actually want to take advantage of the CDKs and activate them. And I wonder if you've thought about that in your setting.
Jason Hughes has a nice paper in Nature Communications where he does this in brain tumors, where he takes advantage of the fact that these cells are sensitive to PDS, and he uses PDS in combination with other chemotherapies. So does that answer the question?
Yeah, I think it's, it's all going to be kind of empirical, testing what would work in combination, but potentially building off the idea that you've made these cells prone to being sensitive to particular therapies just by having deficiencies in ATRX and DAX, and then mutational alterations, and then activating checkpoint repair pathways and so on.
Yeah, and we've also tried looking at the Broad step map data to see if we can find synthetic lethalities that are associated with ATRX mutation. And so far, we haven't really been able to validate any of we didn't try hard enough, but we haven't been able to validate any of these putative hits that we were getting from Project Drive or Project Achilles with ATRX mutation. But again, maybe that's because of the issue of context, because when you're pulling all of these ATRX mutations out of these data sets or those cell lines, they're coming from many, many different types of tumor. And so maybe the context is really important there again.
Well, this is a great discussion, and Laura and Pawel, thanks for such great presentations, and thanks to all of you for your questions during the talks as well. I think with this, we're gonna give up the mic to so we don't all have to stay too late for the final session. I want to again thank everyone, including our speakers.